Hi, everyone, and welcome. Thank you for joining us and joining all the other participants that are here with us today. My name is Patricia Piatti. I am a Senior Product Marketing Manager at Nanotemper.
And I'm Amit Gupta, Senior Product Manager at Nanotemper, and we are both working on the Dianthus product line.
Yeah, so if you're a scientist working in drug discovery at a biopharma company or a CRO, and you're having difficulties and challenges finding hits or validating your leads, and you feel that you are running out of options, and you kind of have come to the right event. We are going to tell you about Dianthus and as a platform that can get your challenging screening campaigns moving forward, especially and particularly if other methods have so far failed you. And failure most, we're focusing on failure in terms of data that is of low quality, data that can't allow you to make decisions that you can trust and feel confident about, so that you know which hits to advance, which leads are true validated leads, and maybe even further characterization of those interactions like ternary complexes when you're working with PROTACs. We are going to also be joined by our colleague, Katarina Braunger. She's going to lead a tour of the platform and also software, both the control software that you use to set up an assay or an experiment, and also a little bit of the analysis software. Okay, so let's get started. So you're a scientist working in drug discovery, and you know these are very exciting times. Exciting, but also challenging at the same time. Exciting because there are new modalities for therapeutics that are being developed, such as protein degraders. But it's challenging because the targets of interest that have been left, and they haven't been able to drug, they're the more challenging ones, the more difficult, we call them undruggables. So those are the ones that are next on the list, and the ones you're working on, and the ones that, one of the reasons why you're having troubles and you're getting stuck in those campaigns. Now, it turns out that there are typically two main issues that are experienced with hit ID and lead validation campaigns. One is that the current screening method generates poor quality data. And it's poor quality, so again, going back to that situation where you don't trust your decisions. You can't feel confident about making decisions on hits or leads. But sometimes the method just doesn't work. And it's typically on those most important targets, right? The most important targets on your pipeline, those are the ones that are the most difficult, of course. And also the other difficulty is that in order to get results, you have to develop experience and expertise. So someone has to be trained in the lab for a long time to develop that expertise and becomes the expert on that particular assay. And typically also includes long and complex protocols, long assay development that also consumes a lot of your compounds and targets. And that also, of course, is time and money. So all of this, adds another layer of complexity on top of the fact that these targets are difficult to drug, and puts, of course, a lot of pressure on drug discovery and screening teams, right?
Yeah, that's certainly true. I mean, in our function as product marketing manager and product manager, we interact a lot with scientists around the globe on a daily basis who work in drug discovery. And in all of those conversations, you often see the same stories emerging or the same patterns appear. And one that we have noticed over the last couple of years, and I think that's very apparent in the whole industry, is that screening campaigns get more and more challenging, of course, because of the increased complexity of the target portfolio. And of course, new modalities being added to the toolbox. But it's not only the scientists in the biophysics labs that are feeling that pressure increasing, but it's also other stakeholders in the whole pipeline, like, for example, the chemistry groups who need high quality data to take informed decisions, which compounds to develop further into lead compounds.
Right. So complexity at every level, right? So can you tell us a little bit more about what makes these targets challenging when it comes to screening campaigns, for instance?
Yeah, certainly. So I think there are three aspects to challenging screening campaigns that I feel are most prevalent maybe at the moment. First, it's the emergence, or let's say the strong traction we are seeing for covalent binders. So covalent molecules are, of course, very, very interesting to drug discovery scientists. But the thing about studying the binding of covalent molecules is that as soon as they bind to the protein of interest, that protein of interest is modified, right? Mm-hmm. So let's say you are working on a sensor surface, that would mean that after the binding of a covalent ligand, you would have to regenerate your protein surface. And that's often, very time consuming. Mm-hmm. Then next, and that's probably the most important point, is that even if you find an assay technology for a given target protein, it does not mean that you necessarily get great data out, right? But the data quality is essential to take informed decision, right? In the end, you need to trust your data in order to take the right decisions to move the screening campaign forward. So it's all not only about finding an assay that works, but also one that gives you great data quality. And then finally, it's the target proteins overall that increase in complexity. So, for example, there are many target proteins that are multimeric proteins, right? For example, E3 ligases are heterooligomers that are certainly very challenging to study. But then to add another layer of complexity, they also engage in multivalent interactions, right? So an E3 ligase, for example, binding to a target protein and a PROTAC on the other hand. So, the whole situation in vitro is getting more complex and requires very sophisticated methods.
Absolutely. So it's more and more complicated from every angle you look at it, right? So it's no surprise, it's not surprising then that you're feeling like, what do I do? I'm running out of options. So like I said initially today, we're here to show you that there is another option that can get you unstuck in a way. And that is Dianthus, right? It's our screening platform, a platform that comes or provides you with not one, but two biophysical modalities, Spectral Shift and TREC. It performs direct measurements of affinity constants, and it does it in solution and in a format that is very, very fitting of the screening campaign or screenings, which is a 384 well plate format. And you'll see it later today, but it's because of its footprint, you can use it as a standalone if your throughput also allows it. But if you need it for larger screening campaigns with maybe hundreds or thousands of compounds, it is compatible. It can be very easily integrated into many automated solutions. Maybe an automated solution is already set up in your lab. So we're talking about liquid handlers and plate feeders as well. So because I'm sure as scientists, you want to see data, you want to see the proof of Dianthus solving these three roadblocks that we described earlier. We have prepared some data for you. And can you get us started on that?
Yeah, sure. So let's focus first on the mentioned covalent binders, right? So the first example we brought you is one that's probably familiar to almost anyone in the drug discovery field. It's the binding of an active compound from the drug Paxlovid, developed by Pfizer, to treat coronavirus patients. And this molecule is a molecule that binds covalently to the main protease of the coronavirus. That's 3CL-Pro, or sometimes also called MPRO. And if you look at that data here, you will first notice the high affinity, which is, of course, expected for a market release drug. But then also you see the high data quality that we can obtain for a covalent binder. And I think the key point here is that all of this is done in solution. We do this in the 384 well plate in small volumes, so meaning there's no regeneration of any surfaces needed. You can study, in row 1, you can study the first covalent ligand, and then in the next row, the second, and you will only consume very, very minor amounts of sample. Then, as mentioned, the data quality is essential to move screening campaigns forward, right? And we brought you a couple of examples to show you the high data quality that you can expect from Dianthus, even for very challenging targets. And one notoriously challenging target that probably many of you are even working on is KRAS. a very frequently studied protein in cancer research. It's has been described as being a protein that's very challenging to work with, right? And what we want to show you here is how for that undruggable, very crucial cancer target, you can obtain high data quality results on the Dianthus platform. Now, in this example, we see the binding of KRAS to a VCB E3 ligase complex. And for this particular experiment, we pre-incubated KRAS with a covalent PROTAC molecule, LC2. And you see the binding of that preformed complex to VCB here with a very high data quality and high signal to noise ratio. And showing this very high affinity means that, this covalent PROTAC results in an efficient degradation of KRAS. Then next, again, a protein that has been described as difficult to drug and difficult to study, STAT3. It's a transcription factor, and that means it doesn't have really biochemical activity. So it does not have any enzymatic function, meaning you need biophysical tools to study small molecule binding to this protein. And that's where the complexity starts, right? I mean, you already limit your toolbox, and then you need one tool that delivers high data quality. And so does Dianthus in this case. You can see the high affinity binding of a PROTAC molecule again, SD36 to STAT3. And please notice the very high data quality again for this particular assay with a signal to noise value of more than 30. And then last example for this section is BRD2, a bromodomain protein, again, a cancer target, and a PROTAC binding to it, MZ1. And in this particular instance, we did not calculate the averages, but we show you the data points of the 3 individual repeats of this experiment. And that shows you how reproducible really the data is, and that you get really stunning data out for these types of interactions. All right, and then finally, we mentioned the increased complexity of the situation when you have multivalent binding for multi-protein complexes. And we brought, I think, one particularly interesting example for you to look at. And that is the formation of a ternary complex of the aforementioned BRD2 protein with the PROTAC MZ1, and now, in addition, an E3 ligase complex VCB. Now, the blue curve is the data that we showed you before. It's the one-to-one binding of the PROTAC and labeled BRD2. But now we add the ligase as the third molecule, and the complexity suddenly dramatically increases, right? We can do this assay again in solution, right? That also means that we can carefully control, for example, the ligase concentration. The ligase is a multi-protein complex, so you want probably to carefully control its concentration in solution. But then you see also that when we add the ligase to the mix and we titrate the PROTAC, that we don't form binary complexes, but in this case, ternary complexes. So we reach a point where we have a lot of ternary complex formed, and then when we add more PROTAC, we see a decrease in the curve again. That's the onset of the hook effect that researchers are really interested in that field, because when you reach this point, then the binding sites on both proteins get saturated and no ternary complex can be formed anymore. So you see how the high data quality of Dianthus that you can obtain for this very complicated system allows you to match it to the latest mathematical models that can describe these 3-body problems.
So all this fantastic data was obtained from a Dianthus using Spectral Shift. So Spectral Shift as a concept is not new. It's been used before. What is novel here in this case is how it's used to derive affinity constants. So can you tell us a little bit about how it works in Dianthus, how we do that? Yeah,
definitely. So this is actually how Dianthus is powered, right? It's how it works under the hood. We use a fluorescence readout, as you might have already guessed from the previous slides. One of the components in the mix needs to be labeled. Usually that's the protein of interest. And you can see here on the slide how a labeled protein, and we label with small organic fluorophores usually, exhibits a fluorescence emission spectrum in the red spectral range. Now, on the right-hand side of the slide, you can see how that emission spectrum can be affected when a small molecule binds to the protein. So when a small molecule binding to the labeled protein occurs, then that small molecule will have an effect on the chemical microenvironment of the fluorophore. That chemical microenvironment of the fluorophore is affected either by proximity of the small molecule or by the small molecule inducing a conformational change. And since the environment of the fluorophore changes, that has an effect on the fluorescence properties, such as the emission spectrum. And you can see here in this illustration how the emission spectrum can either change to more red wavelengths or more blue wavelengths. However, even this is sort of an exaggeration. So most of the spectral shifts that we are measuring are sub-nanometers, so very, very subtle shifts that require highly sophisticated instrumentation. Now, by focusing on two wavelengths, 650 and 670, we can calculate a ratio metric value that tells us about the shift in the emission spectrum. And using that approach, this ratio metric approach, would basically result in data that is unbiased by pipetting errors, for example. So it's not dependent on absolute intensities. And plotting that ratio value against the titrated ligand concentration will result in these characteristic sigmoidal binding curves, which we can then use to fit them with the law of mass action and derive affinity constants, dissociation constants, and so on.
Great. So sophisticated optics and robust hardware is important to get this kind of data. But also we mustn't forget about having good consumables and reagents, right? They play a very important role as well in your assays. So in this case, in the case of Dianthus, you have the 384 well plates and the labeling kits. So the plates, you will learn a bit more details during the tour of Dianthus, but just to tell you that we're designed specifically to measure whether both spectral shift and trick measurements. They have a unique trackable barcode. And they also, the wells of the plate have been coated so that to reduce protein absorption to the walls of those wells, because you're going to, in a lot of cases, work with very low concentrations of your target and also low volumes. When it comes to the labeling kits, you have a choice of many chemistries, meaning where that fluorophore will be added in the molecule that you're labeling. The protocols are typically, they're all very simple to perform, very few steps. And that also means that they don't add time to the overall time that you need to perform the assay. And also come in different sizes. So if you have just a few experiments to perform, so you need to label just a little bit of your protein target, you have a low volume kit. But if you need to label larger amounts, then you have a large volume labeling kit as well. With this, let's get really into now the tour of the Dianthus. Katharina is now ready to show you both the Dianthus and the software. So enjoy the tour.
Thank you, Patricia. My name is Katharina Braunger, and I'm a product specialist at Nanotemper Technologies for Dianthus. Over the next few minutes, I'd like to give you a brief tour on the instrument. I'm going to show you a little bit about the instrument platform, and then also I'm going to talk about how the Dianthus software can support you in moving your challenging screening campaigns forward. So let's get started. Dianthus is a biophysical instrument which provides high-quality interaction analyses for affinity and single-dose screens. It measures your samples in solution in 384 well plates using low volumes and low concentrations of sample. You can use Dianthus as a standalone benchtop instrument. However, you might also choose to integrate it with your existing automation solutions, such as liquid handlers or plate feeders. Once you switch on the instrument, it's ready to go. So there is no rinsing or washing required, since there's no microfluidics in the instrument. The Dianthus plates are specifically designed for the fluorescence-based spectral shift and trick measurements. They have black walls to avoid any crosstalk between neighboring wells. The clear bottom provides a window into your samples for fluorescence excitation and detection. Since Dianthus uses very low protein concentrations, the inside of the wells is coated to prevent adsorption. Also, each plate has a unique barcode so that it can be recognized by the instrument. If you are running larger screening campaigns, you can also choose to seal the plates using either heat or glue sealing. The stage on the instrument is operated from the control software on the computer. Once a plate sits inside the instrument, it's located above a highly sophisticated optical setup which provides reproducible and precise measurements. So now I'm going to go into the software to open the gate and then place the plate inside the instrument, and then I'm going to show you how you can really quickly set up an experiment in the Dianthus control software, and also give you a quick glimpse at the Dianthus screening analysis software and what it looks like for a single dose screen. So let's go into the software.
We're now in the Dianthus control software. The panel at the bottom is used to control the instrument. For example, opening and closing the gate, queuing an experiment, or starting the measurement queue.
As you click the corresponding button in the control software, Dianthus opens the gate and presents the stage which holds the plate during a measurement. Plates can be placed manually or inserted by a plate feeder. Another click in the software closes the gate.
As you can see, Dianthus control software has different experiment templates for assay development, single-dose screening, and affinity screening. When you hover over the name of a template, there's a preview which gives you more information on what you can do with this type of experiment and when it might be a good choice for the assay that you're planning. Now I'm going to show you how you can set up an experiment with just a few clicks. Let's say you'd like to measure an affinity series. Then the binding affinity template is perfect for this. When clicking on the first well to be measured, the software automatically creates a 12-point dilution series. You can easily edit your experimental parameters here. For example, you can change the names of the interaction partners or adapt any of the concentrations that are being used in the assay. In addition, you can choose whether you would like to run a spectral shift only experiment or include trick in your measurement. Once you're happy with the settings, you queue the experiment in the bottom panel. What if you're running a screening campaign? With the import templates, you can upload any plate layout from an Excel spreadsheet. This way, even when running multiple plates in a row, it takes less than a minute to set up your measurement. Now I'd like to go to the Dianthus screening analysis software to show you what a single dose screen will look like. The screening analysis software has 4 tabs where you can select the data to be analyzed, choose the references for normalization, get detailed information on all ligands in your screen, or see a summary of your campaign. The Ligands tab is where you can dive deep into the data. Here, we're looking at a fragment screen with just under 3,500 data points. The scatter plot on the left gives you a quick visual overview of all the data in the screen. The table, as well as the data panels on the right, provide detailed information for each individual ligand. The software uses smart algorithms to automatically assign ligands to different categories, reflected by the color coding in the scatter plot. For example, non-binders are shown in gray, and hits are displayed in purple. In addition to detecting hits, the ligand classification also automatically identifies molecules that can cause artifacts such as aggregation or autofluorescence. Let's take this ligand for example. As you can see, it's in the autofluorescence category. Taking just the response, it might be considered a hit, and also the two replicates overlap very nicely. However, the initial fluorescence for this ligand is significantly higher than for the corresponding DMSO reference, meaning that the response could also be attributed to a fluorescence artifact. Or let's take this ligand, which has been flagged as inconclusive due to well-scan anomalies for both replicates. Bumpy well scans can be a result of air bubbles or dust particles, but they are also a hallmark of aggregation-inducing molecules. A trick experiment on Dianthus is a quick and great way to distinguish between these different options. In order to be classified as a hit, a ligand has to pass all the quality checks and show reproducible behavior between the replicates. If you disagree with the classification, you can always manually overwrite any quality check in the software. The Summary tab has a pie chart showing you exactly the proportion of ligands in any given category. You can select categories to inspect them in more detail in the Ligands tab, or you can export your data for reports or further analysis. I hope that you found this quick tour insightful, and thank you for your time.
Okay. Welcome back. We hope that you enjoyed the tour of the instrument and the software. And before we move forward with the rest of the event, I'd like to summarize the few of the topics that we discussed today. So where you can really rely on Dianthus for your challenging screening campaigns. So we talked about the characterization of covalent ligands, and we told you about how important in this case is to make the measurements in solution, where you don't have to immobilize the target to a solid surface. So it's less complicated, stress-free and more budget-friendly, because it's a lot more affordable not having to use fresh biosensors every time or for every of the compounds you need to analyze. The other one was methods that give you noisy and unreliable data. And you've seen with the use of the Spectral Shift what fantastic data we're able to produce, and high signal-to-noise ratios that are above and superior to other technologies that you may have tried. And third is when you need to characterize interactions that involve multimeric molecules or multivalent interactions. We showed you an example of PROTACs, which is a pretty complex situation. You have multimeric ligases and then three molecules coming together. So there's a lot going on. And again, having measurements in solution with flexibility of how you set up the assay and with respect to which molecules you're going to label also, is the case for Spectral Shift. And then at the end, you get data that agrees very nicely with these latest models that describe the behavior of these three component interactions. So I think we're then ready for the Q&A session. And before we get into it, please don't forget to visit the Event Resource Center. You'll be able to download some collaterals and also have some links to other pages that give you more information about the topics that we are discussing today. And during the Q&A, we're going to try to answer as many questions as possible. But if we don't get to answer yours, we're going to make sure to reach out to you and address your inquiries. All right. So let's see what we got in the chat throughout the event. All right. One about the licenses, software licenses. How many software licenses do you include with each instrument? And do I have to purchase the analysis software separately?
Yeah. So with each instrument that we ship one license of the control and the analysis software. So that means that the control PC in the lab is fully equipped to run and analyze data. However, sometimes you might want to use additional software licenses on your PC in your office because it's more quiet, for example. Then we offer packages of 1 license or packages of 5 licenses for each control and analysis, so that you are up and running and you can equip, for example, a whole team to work very efficiently with the data that you obtain.
Right. Another one related to software. Are the files exported from the software compatible with gene data? I don't know if this relates to just the analysis software or the control, but
yeah. Well, in essence, both softwares have a very comprehensive data export. So when you export data from the software, you get a file that contains not only the analyzed affinities, but also the raw data that was measured by the instrument. And that means those files can be easily used to upload into any or import into any third-party software for, let's say, further data analysis or data storage.
Okay. Okay. This one is about spectral shift and trick. How do you measure spectral shift and trick? Do you need 2 sets of samples, one for each measurement?
Yeah. Excellent question. So the instrument, when you select to run both spectral shift and trick, that's, by the way, done by a simple click of one button. The instrument would obtain the data for spectral shift on one well, and then right after do the trick measurement as well. So that way you get the data from the same specimen and measured at fairly the same time. So that would allow you later on the correlation between the 2 data sets that you get.
The perfect kind of type of replicate, I guess,
exactly.
you could get, right? Are the wavelengths used by the instrument fixed, and can you use other fluorophores?
Yeah. So yes, the wavelengths of the instrument are fixed for 2 reasons. So the first reason is that when you work in the red spectral range, you have very few compounds, small molecule compounds, that show autofluorescence in that range. So you get less artifacts. And then there's a second reason here. As I mentioned, we usually measure very subtle shifts in the emission spectrum, sub-nanometer. And to do so in a reproducible way, you need fixed wavelength filter sets, like we have in this case, to really achieve that type of precision. Having said that, you can use any fluorophore that's in that spectral range, but also I would like to caution you that there are some fluorophores that are not very sensitive to changes in their microenvironment, right? So those fluorophores are probably much less suitable for this kind of an assay.
That is probably even more important than the wavelengths, right? That to be in that right range is that they need to be sensitive to these changes, because that's what's going to cause those shifts in the emission spectrum, right? All right. Another one about spectral shift and trick is, when would you choose to use both or measure both spectral shift and trick?
Yeah, we get that question a lot, right? I mean, it's in essence, it's a question about why having 2 modalities in one instrument, right? And I think there are fantastic examples where one method, gives you additional information. And those are cases where aggregation causes problems, right? So let's say you are looking at a small molecule that would induce aggregation of your target protein. In those instances, it can be very useful to measure trick as well, because trick is a method that's quite sensitive to that and can actually show you if your protein is aggregated or not. So sometimes I've seen cases where there's conflicting information about the small molecule. So one method says it binds, one method says it does not bind, and where we then see in trig assays, oh, well, that compound induces aggregation of the protein. And there you have that discrepancy explained right away. But then there are also instances where you don't want to be so susceptible to aggregation in your method, right? And that's what Spectral Shift delivers. So let's say you have an assay for an IDP, which is intrinsically more aggregation prone. And then basically you have to deal with what the protein is, right? That it has those aggregates in the mix. Now, in those instances, you still want to screen against those targets, right? So you need a method that delivers you robust binding data, even in the presence of a fraction of aggregates. And that's the strength of Spectral Shift.
It's almost like having a QC, a built-in QC system, right?
Yeah,
with
absolutely.
in those instances.
Yeah, definitely. Trig can be considered kind of a quality control measurement included in your database.
Right. In the same platform, you have both. Yeah, that's great value, I think. So looking at the time, we have time for just one more question.
Right.
Can you use other fluorophores besides the ones provided in your labeling kits? And
So I went into that already a little. So as said, the spectral range should match, and it should have some sensitivity to the microenvironment. One fluorophore that I've seen used often, apart from ours, of course, is Cy5.
Yep.
And it's often used in instances where you, for example, working with a pre-labeled DNA oligo, because the vendors of those oligos, they usually can already attach a Cy5 during the synthesis.
Right. Okay. Great. Thank you. Okay, we've reached the end of the event with a Q&A session done. And so thanks again for joining us. And I hope you leave with the notion that you're not out of options. If you're challenging, your screening campaigns are kind of stuck and halted and you can move forward, that there is hope. There's another solution out there and you should give it a try. And again, thank you for joining us.
Yeah, thanks a lot. We hope that you enjoyed this session and maybe talk to you anytime soon. And we just wish you a wonderful rest of this day or evening or night, wherever you may be.
That's right.
Bye-bye.
Bye-bye.